Our next speaker is, I think, John. He's going to talk about a proteomic profiling approach uncovers early markers of Alzheimer's disease linked to neurotoxicity. John.
Thanks. Okay, so how do I do this? So I'm very happy that the previous speaker actually talked about AD, then it saves me some time. So I hope this works. Okay, so my lab is actually very focused on the synapse itself, and by synapse I also mean neurocircuitry. So the reason why I show this lab, there are two reasons actually. One is to celebrate the serendipity of science, because we started off with trying to identify basic mechanisms in synapse formation and neurocircuit function. We ended up in AD, and this is a topic that I'm going to talk about for the rest of the talk. So as I mentioned, I'm very grateful to the previous speaker, so that I will skip all this part of the slide. Just to mention to you that AD is a progressive neurodegenerative disorder, and there's no disease-modifying therapy currently available. So it's actually a very bad thing, because more and more of us are growing older, and more of us have the possibility of suffering from the disease. Now, if you look at AD itself, we know that there's a big issue, because most of the putative cures that have been out there have not been successful, and I think there's a need to better understand, what causes the disease and how we can help to ameliorate and even cure the disorder itself. There are two ways of looking at it, I feel. One way is to have this gene-centric way of looking at it, and this has been very helpful in understanding the disease. For instance, if you look at the autosomal dominant AD genes or the familiar AD genes, these are the major drivers over there. And if you look at the sporadic forms of AD, there has been an increasing number of genes that have been identified that contributes to sporadic AD itself. Now, while this is very helpful, and I cannot overemphasize its importance itself, I think there is also a growing opinion that while you can have a genetic driver, so for instance A, beta, and tau that drives synaptic loss, neuronal death, and cognitive decline itself, there is an ancillary set of pathways, parallel supporting antagonizing pathways that is contributing to the primary effect caused by the primary gene drivers itself. And to try to better understand this, there has been a number of diagnostic techniques to try to better define or rather to stage the patients to better help identify patients who might be at risk of AD itself. And you can see that with technological development, there are increasingly sensitive tools to help us to do this. But I think there is better thing, we can do better in trying to identify within this window precipitating events that could help us stratify even better who might already be suffering from AD at a pre-asymptomatic stage, and also to identify new mechanistic insights to help us look at or to understand how AD is generated. So a couple of years ago, what I'm talking about is a recently published book, and it's driven by two very talented graduate students, Sasha and Sarah. Sasha just graduated last year. Sarah is still continuing his work with us. So Sasha started off with the project, and the idea was actually very simple. We want a very unbiased approach to look at what happens in AD, and what Sasha did at the time was to take 3 times TGAT beta animals, or wild-type control animals, and we started to stage them at the 2, 6, 12, and 18 months. And this corresponds roughly to the asymptomatic at 2 months. You might say this is MCI, and this is the overt AD stage itself. So what he did was to extract the brain, divide it into 2. One half, we analyzed it by quantitative mass spectrometry. The other half, he saved it so that we can do immunohistochemistry subsequently for targets. For the MS, I'm not going to go through all the details, but essentially, after we filter through the data, we have the statistically significant up-down regulated proteins. We end up with a number of proteins that we can analyze in a variety of manner. There's a lot of interesting data over here because it also addresses aging, but I'm going to focus more on AD itself. And to crunch all the data, you can do something what we call a principal component analysis. And if you crunch all the data, as I mentioned, you can differentiate AD causing proteins that are changed, or aging proteins, or proteins that are related to the aging that are changed in the brains of these animals as we go along. So I'm going to focus on the early asymptomatic markers because this is really where our interest is to try to identify early symptoms. And one protein that I'm going to talk about more is this protein known as HEBP1, as you can see is right at the top of the principal component analysis itself. Now, I mentioned this protein as well. These are the volcano plots of the proteomic analysis, and those of you who are not familiar with this, anything to the right of the plot is proteins that are upregulated. Anything to the left of the plots are proteins that are downregulated. And of course, the red little spots over here are all those that are significant. Okay. So HEBP1 is very interesting because you already see that there's very high and very significant expression at the asymptomatic stage. Now, 2 months, 6 months, 12 months, and 18 months, you can see the HEBP1 is consistently upregulated. So it's very interesting because now we have a protein that we can identify at the asymptomatic stage of AD itself that might be contributing to the early part, to the early phase of AD. Now we validated this in first in the mouse model. We took brains from 2-month-old wild type and 3 times TG80 mice, and you can see that HEBP1 is consistently upregulated in comparison to controls itself. Now you might be asking, okay, fine, this is the mouse, what about humans? So we were very lucky to be collaborating with our clinical partners, and even though they gave us a very small samples data set, but what we could already see is that particularly in the aggressive forms of AD itself, HEBP1 is significantly upregulated as compared to controls. Now, as I mentioned, this is a very small data set, and we really want to understand whether, in the broader context of AD, is HEBP1 relevant? So what Sasha did at the time was to look at publicly available databases, and you can see if you look at the mRNA expression profile of HEBP1 in the prefrontal cortex and visual cortex, for instance, the more AD-relevant brain regions, you can see that the mRNA expression levels of HEBP1 is very highly elevated. So again, attesting to the idea that HEBP1 is relevant to human AD itself. So HEBP1 is very understudied, and nobody really knows what it's doing in the brain. So we started initial characterization, and we were able to confirm again that in the brain regions, in various brain regions, including the cortex, hippocampus, cerebellum, and brainstem, that HEBP1 is significantly upregulated. Now, is it a neuronal protein, or is it microglial astrocyte? The IHC data that you see over here would indicate that HEBP1 is very highly expressed in neurons and not so much so in GFAP and GFAP positive astrocytes or EBA1 microglial cells as well. Now, if you look at the histology, because I had a discussion with Morgan Zhang, and he was saying, you have to look at microglial, and I say, okay, fine, I will look at it. But if you look at this, this is the 3 times DG80 brain itself, and this is the control brain itself. You can see that the upregulation is actually in the neuronal regions itself. So this is the, for those who are familiar, the CA1 of the hippocampus itself, again at the cell body layer, you see only upregulation of HEBP1 in neurons. So what is it doing in neurons? Actually, nobody really knew. So we started to overexpress the proteins. We had some problems with the antibody itself for this. So we overexpressed the protein, and we saw that HEBP1 localizes to the mitochondria. And you can see this line scan analysis of HEBP1 tagged proteins as compared to MitoTracker. We confirmed this in biochemical studies using purified mitochondrial preparation. So the work over here is from now on is contributed by Sarah. You can see the HEBP1 is present in mitochondria and not present in the supernatant in its preparations. So HEBP1, its full name is called heme binding protein 1. And when Sasha told me about this, I was saying, well, what does that really mean? Because we are looking at neurons itself. But as it turns out, even though the protein is very poorly recognized, it binds heme and the precursor of heme. And we know that heme metabolism is dysregulated in AD. And we know that the A-beta heme complex induces oxidative stress, thereby linking it to mitochondria itself. Now, I'm not going to go through this in detail, but the heme component is extremely interesting as well, because we know in AD patients there are people with damaged vasculature. So I'm going to go very quickly now, sorry. So we looked at what happens in neurons with or without HEBP. So we could knock this out with CRISPR, and you can see that if we expose this to heme, which is a hemine, which is a surrogate for heme, you see that there's increased cytotoxicity in wild-type neurons, but not so much in HEBP knockout neurons itself, indicating that it's specific to exposure to heme. It doesn't get, neurons don't die when you give it other stress. And the relationship to mitochondria is particularly interesting as well, because you can see that when neurons are exposed to heme, there's mitochondrial damage, and interestingly, even more surprisingly, HEBP1 knockout neurons have even more mitochondrial damage itself. But if you look at the survival, then one questions why the neurons don't die, and we started to look at the apoptotic pathway. And what I'd just like to point out over here is that in heme-treated cells, like in control cells, we know that the mitochondria is damaged because cytochrome C comes out into the cytosol, again, supportive of the idea that there's mitochondrial damage itself. We went on to show that the apoptotic pathway itself is not activated in HEBP1 knockout neurons, whereas in wild-type and control neurons, the pro-caspase-9 is cleaved into caspase-9. We show that the caspase-3/7 cleavage is much higher in wild-type as compared to HEBP1 knockout neurons itself. Now, this is my very last slide. So we were asked to do this by reviewers, and they were saying, you know, well, okay, fine, show me HEBP1, but what about A-beta itself? And this is very, very amazing. So you can see that neuronal death with hemine is about 60%, with A-beta is about 40%. You add the two together in wild-type cells, you reach almost 90%. Now, if you knock out HEBP1, neither hemine, A-beta, or the combination of A-beta and hemine itself causes death in HEBP1 defective neurons. So it's very nice, and I'm very glad that the reviewer actually asked us to do this. So in summary, I didn't have time to go through all the changes, but we know there's a lot of massive remodeling in the brain protein in AD itself, and the HEBP1 is a very interesting and potentially important, not just biomarker, but protein that's involved in AD pathogenesis itself, whose mechanism probably hits at the level of neurons. And with that, I'd like to thank you for your attention. I'd like to thank my two very fantastic students, Sasha and Sarah, who has done a lot of the work. Sarah has a postdoc in EV71, and Yinghua, my other very talented graduate student, will give a talk tomorrow on brain organoids and so on. With that, I would really like to thank you. I'm sorry for exceeding the time.
I think we can still have questions. I really want to have a question coming up. Yeah, Tongwei.
Which subcompartment of mitochondria is HEBP1 found?
Yeah, it's very interesting. If you look at the IF data, it's actually present. It's attached to. It's not an integral part of the mitochondria. It's attached to the mitochondria. So we can talk later on why. Yeah.
Very nice talk, John. So a couple of questions. So one is about, so you started off saying that you wanted to look at early markers for AD, even before the symptoms start to show up, probably. Now, what from the data you're showing, it would seem like this is probably towards the later aspect when you start seeing cell death. Is that what you think, or is it like, how do you connect these two aspects of how you started versus?
Yeah, so that's a very good question. So when we started off with this, we thought we could identify mechanisms that already prime the brain, that tells you something's not going right. And actually the earlier question on that Suchun brought up is sitting on my mind for a very long time. What happens, So what was surprising for this, and actually I had a very interesting discussion at Eddie as well, and he was saying, this thing is coming out so early, what's it doing? But if you look at the data that we have seen, my guess is still a guess, is that at a very early stage, there is already something causing the brain to know that it's damaged. And the question is always, is very interesting. Why is HEBP1 neurons dying? So is it a protective mechanism or is it a pathological mechanism? So my guess at the moment is protective. So at the early points in time, it's actually protecting the brain by actually removing neurons that are slightly damaged. But I think you're probably familiar with the idea that AD patients have a lot of vascular damage as well. So this is where I think the program becomes hijacked by pathology. So as the vasculature damage becomes more and more pervasive, and the A-beta complex itself becomes even more and more important in causing oxidative damage, then the system takes over, and there you start to see massive neuronal death. So I think it's a two-step mechanism. One is the body's desire to be protective, but that itself is ultimately hijacked by the pathology itself. Yeah. So at least that's what we're actually testing some of these ideas now, but that's the current idea that we are testing.
Okay, you can speak out.
Hi, great talk. Is there a differential expression of the protein you're working on between between excitatory and inhibitory neurons?
Yeah, very good question. So we haven't looked at that in detail, but at least based on the cursory view, one would almost imagine it's more excitatory. Yeah, but this is just a very cursory look at the immunohistochemistry. Yeah.
Okay, thank you, John. Thank you.